Hello, I'm Martin Blaser. I'm the director of the Center for Advanced Biotechnology and Medicine, or CABM. Today, I have the pleasure of speaking with CABM resident faculty member, Dr. Alexander Valvezan. He's an assistant professor in the Department of Pharmacology at Robert Wood Johnson Medical School. Hi, Alex.
Hi, Marty. Great to be speaking with you.
Yeah, the same. So I just want to start. Can you tell us a little about your early education and what you were doing before you came to CABM?
Sure. So I did my undergraduate at the University of Delaware, where I received a degree in biology. And from there, I went to graduate school at the University of Pennsylvania. And I did my thesis work in the laboratory of Dr. Peter Klein, studying the Wnt signaling pathway in embryonic development and in cancer. And that work actually led to an interest in cancer metabolism, which I pursued during my postdoc at Harvard University in the lab of Brendan Manning, studying cancer signaling and cancer metabolism with a focus on the mammalian target of rapamycin complex 1, mTORC1.
Right. So how long have you been at CABM now?
I started just in September of 2020, so just a few months ago.
So you're a newbie,
That's right.
and so you're setting up your lab. What's the main focus of your lab now?
We're focused on understanding how cellular metabolism is regulated in cells that are growing and proliferating both under normal conditions and in the context of diseases such as cancer. So we know that the metabolism of cancer cells is drastically different from that of normal cells to support its rapid growth and proliferation. But really what those differences are and how they come about is an area of active investigation. And that's one that my lab focuses on. And in particular, we're interested in understanding how we can use that information to identify and exploit vulnerabilities in tumors.
That seems very worthwhile. So how are you approaching it?
We use a combination of genetic and pharmacological techniques to study gene function, as well as mass spec-based metabolomics and metabolite tracing approaches to study cellular metabolism. And through combining these types of techniques, we hope to gain a comprehensive overview of how particular signaling pathways or particular oncogenic mutations influence cellular metabolism. And then we use that information to apply it translationally in order to identify new therapies.
So what's one new project that you're working on that you're particularly excited about?
Well, I would say there are a number of projects that I'm excited about. Being at this stage of my career is pretty cool because there are a lot of interesting directions to go in. But one that I'm particularly interested in and excited about builds off of work that I did as a postdoctoral fellow, where I discovered a targetable metabolic vulnerability that arises downstream of the protein complex that I mentioned earlier, mammalian target of rapamycin complex 1, or mTORC1. And essentially what I found is that mTORC1 confers a dependence on certain nucleotide synthesis pathways that we can then target for anti-tumor effect. And that work was done primarily in cell and tumor models of a genetic tumor syndrome called tuberous sclerosis complex, which is driven by high mTORC1 activation. But it turns out mTORC1 is not just activated in that tumor syndrome, but it's also activated in the majority of human cancers across nearly all lineages. And so what I'm particularly excited about is really to determine how widespread this vulnerability is and whether there are additional settings, particularly cancer settings, where we can apply this type of strategy for therapeutic benefit.
And so how will you find those other cancers?
Through a combination of cell line screening, as well as bioinformatic approaches, and in particular, screening particular oncogenic mutations that we know are very common in cancers and are direct upstream regulators of mTORC1. So we know that those mutations strongly activate mTORC1, and therefore we predict that this vulnerability might be present in those types of cancers. And those include mutations in the PI3K, AKT pathway, mutations in the RAS pathway, and other very prevalent mutated pathways.
Great. That's exciting for me too. So how do you interact with students or trainees? I guess based in your past labs and then in your future lab. What are your goals there?
My goals are to strike a balance between providing guidance and direction while also allowing my students and trainees the freedom to pursue their own interests and make their own mistakes. I believe that this is certainly true for me as a student and a trainee, that people will be most successful when they're pursuing something that they're passionate about. So I hope to, when someone is interested in my lab, rather than saying, okay, I'm going to require you to work in this particular area, I hope to really identify areas that they're interested in, that the lab, that overlaps with the interests of the lab and that the lab is suited to pursue, and really outline a path forward for them that really motivates them.
Great, great. Now, I know you just arrived at Rutgers. Do you have any other roles at Rutgers that you want to communicate?
I'm a member of the Cancer Institute of New Jersey.
Mhm. Now, just before we end, I want to ask you a question that's a little unusual, and that is, and especially for somebody as young as you. Now, imagine we were writing your scientific obituary, and we had to say, well, he was a great scientist because. What would that? How would you like to be remembered?
Hopefully it's not something we have to think about too soon, but
That's
I think in the long term, I think there's really two aspects. One is the type of science that I do, and the other is the people that, the impressions that I leave on the people that I've interacted with. I hope to leave my trainees with the same passion for science that I had coming in, that really motivated me to do what I'm doing. And then in terms of the type of work that we do, really just things that I hope are to do work that has a solid foundation that stands the test of time. Of course, I want it to be important and impactful. I recall a perspective written by Bill Kalin, and I believe it was in Nature a few years ago, but it really resonated with me, which was, the title was something like, publish houses of brick, not mansions of straw. And I thought that's very important, is rather than kind of these, expansive, all-inclusive types of studies that sometimes may not have solid foundations, to really make strong advancements that stand out.
Yeah, that's actually, I hadn't seen that paper, but it's interesting because for many years I've talked about when you do an experiment and you get a result, I call that result, it's a brick. And you build your house by building bricks. And the great thing about science is that one of the bricks that you make, I can use in my house and vice versa. But the importance is that the bricks be solid,
That's
because
right.
if they're straw, we're in trouble.
Yeah.
Great. Okay. Thanks so much for talking to me.
Thank you.